Chapter 11. Absorption and Transport. Absorption is the stage where the products of digestion move from the intestinal lumen into the body's internal environment, but this transfer depends entirely on the form in which those products arrive. The intestinal lining is structured to take up specific molecular forms through defined transport mechanisms. Amino acids, small peptides, simple sugars, and lipid transport structures are handled through pathways that require prior reduction and organization. When digestion has proceeded with proper sequencing, these forms are present in consistent quantities, and absorption operates as a continuous, efficient process. When upstream breakdown is incomplete, the available substrates do not match these transport systems, and the movement of nutrients across the intestinal barrier becomes limited and uneven. The surface of the small intestine is organized to maximize contact between luminal contents and absorptive cells. This structure allows for high-capacity uptake when substrates are in their intended forms. Transport proteins within these cells move nutrients across the epithelial layer, and this movement depends on both concentration gradients and the availability of compatible substrates. When peptides and amino acids are present in sufficient amounts, protein-derived nutrients are taken up efficiently. When carbohydrates are fully reduced to simple sugars, their transport proceeds rapidly. When lipid-derived components are organized into micelles and related structures, they can approach the epithelial surface and be absorbed. The transition from the lumen into the cell requires that substrates be both chemically and physically prepared. Larger fragments or poorly organized molecules do not engage transport systems effectively. They remain within the lumen or move forward without being absorbed. This creates a situation where the presence of nutrients does not translate into uptake because the structural requirements for transport have not been met. The efficiency of absorption is therefore determined upstream, at the level of digestion, rather than at the level of the transport mechanism itself. Once nutrients cross the epithelial layer, they enter distribution pathways that deliver them to different regions of the body. Water-soluble components move into the circulation, while lipid-derived components follow a pathway that reflects their hydrophobic nature. These transport routes depend on the initial form in which nutrients are absorbed. When absorption is consistent, distribution proceeds with predictable patterns, supplying tissues with the substrates required for energy production, structural maintenance, and signaling. When absorption is uneven, distribution becomes variable, and the availability of nutrients at the tissue level fluctuates. The coordination between absorption and transport also depends on timing. Nutrients must be absorbed in a pattern that aligns with their arrival in the intestine. When gastric emptying and intestinal motility are consistent, substrates are presented to the absorptive surface in steady flow, allowing transport systems to operate continuously. When upstream processes are irregular, substrates arrive in pulses or in forms that vary in readiness for absorption. This disrupts the continuity of uptake and introduces variability into the transport phase. Absorption is therefore not a passive endpoint, but an active process that reflects the entire sequence that precedes it. The structure of the intestinal lining and its transport mechanisms are designed to operate on substrates that have been properly prepared. When this preparation is achieved, absorption and transport proceed with stability and efficiency. When it is not, the system encounters substrates that it cannot fully utilize, and the movement of nutrients into the body becomes constrained by the conditions established earlier in digestion. Membrane transport systems. Membrane transport systems determine which digestion products actually enter the body and at what rate, and they operate with strict structural requirements. The intestinal epithelium is not a passive surface that absorbs anything present in the lumen. It is a selectively gated interface built from lipid membranes embedded with transport proteins, each designed to move specific molecular forms across the barrier. These systems recognize size, charge, and configuration. When substrates match these parameters, transport proceeds efficiently. When they do not, the substrate remains in the lumen regardless of how much is present. Protein-derived nutrients are absorbed through transporters that move amino acids and small peptides across the epithelial membrane. These systems require proteins to be reduced to a defined range of sizes. Dipeptides, tripeptides, and individual amino acids are compatible with transport mechanisms that move them into the cell. Larger fragments do not engage these systems effectively. They remain outside the membrane, continuing along the intestine or interacting with the epithelial surface in ways that can trigger secondary responses. The limitation is not the number of transporters. It is the structural form of the substrate presented to them. Carbohydrate transport follows a similar pattern. Specific transporters move simple sugars across the membrane, relying on concentration gradients and cellular energy to drive uptake. These transporters do not process complex carbohydrates directly. The substrate must be reduced to monosaccharides before it can be absorbed. When breakdown is complete, transport is rapid and consistent. When reduction is incomplete, fragments that cannot engage these transporters persist within the lumen, contributing to variability in both absorption and downstream microbial activity. Lipid transport introduces an additional layer of structural dependency. Fat-derived components must be organized into micelles to approach the epithelial surface, and once inside the cell, they are reassembled into transportable forms that can move through the body. This process depends on the prior action of bile and lipolytic enzymes. Without proper emulsification and breakdown, lipid components do not reach the membrane in a form that can be taken up. The membrane itself, composed of lipid bilayers, interacts with these substrates in a way that reflects their structural compatibility. Properly organized lipid products integrate into this system, while poorly prepared aggregates do not. The membrane environment itself influences transport efficiency. The composition of the epithelial membrane, including its lipid structure, affects fluidity and the function of embedded transport proteins. This structure determines how easily substrates can interact with and pass through the membrane. When the membrane is operating within its intended parameters, transport systems function with predictable capacity. When the internal environment is altered, the behavior of these systems can shift, affecting the rate and consistency of nutrient uptake. Timing and concentration also shape how these transport systems operate. A steady delivery of properly prepared substrates allows transporters to function continuously, maintaining consistent uptake. When substrates arrive in irregular pulses or in mixed states of readiness, transport becomes uneven. Periods of high availability may be followed by periods where compatible substrates are limited, reducing overall efficiency. The system responds to these fluctuations, but the variability originates from upstream conditions rather than from the transport mechanisms themselves. Membrane transport systems therefore act as the final filter in the digestive sequence, enforcing structural requirements that reflect the design of the system. They do not compensate for incomplete digestion. They accept substrates that meet their criteria and exclude those that do not. The effectiveness of absorption is determined by how well upstream processes produce the forms that these systems are built to handle. Chylomicrons and lipid delivery. Lipid absorption does not end at the intestinal membrane. Once fatty acids and related components enter the epithelial cell, they are reorganized into larger transport structures that can move through the body without disrupting the aqueous environment of the circulation. Chylomicrons are the primary carriers that perform this function. They package lipid components into a form that can be distributed to tissues while maintaining compatibility with the fluid systems that transport them. Inside the intestinal cell, absorbed lipid components are reassembled into triglycerides and combined with structural molecules that stabilize the particle. This assembly creates a spherical structure with a lipid core and a surface that allows it to remain suspended in fluid. The formation of chylomicrons depends on the prior steps of digestion and absorption. Lipids must be reduced to forms that can cross the membrane, and those components must then be available in sufficient quantity to be repackaged. When upstream processes are aligned, this assembly occurs efficiently, producing particles that can be released into the transport system. Chylomicrons do not enter the bloodstream directly. They move into the lymphatic system, which provides a pathway suited to transporting large lipid-rich particles. This route allows lipids to be distributed without overwhelming the immediate circulatory environment. From the lymphatic system, chylomicrons eventually enter the bloodstream, where they deliver their lipid content to tissues that require it for energy, membrane structure, and signaling processes. The structure of chylomicrons determines how lipids are delivered. Their size and composition allow them to interact with enzymes and tissues in a controlled manner, releasing fatty acids where they are needed. This process depends on the integrity of the particle. When chylomicrons are properly formed, lipid delivery is gradual and coordinated. When formation is impaired due to incomplete digestion or absorption, fewer particles are produced and lipid distribution becomes limited. The efficiency of this system is tied to earlier stages of fat processing. Emulsification, enzymatic breakdown, and micelle formation all determine how much lipid reaches the epithelial cell in a form that can be absorbed. If these steps are incomplete, the pool of available components for chylomicron assembly is reduced. This limits the amount of lipid that can be packaged and transported, affecting the supply of these molecules to the rest of the body. Chylomicron formation also influences the timing of lipid delivery. Because these particles move through the lymphatic system before entering the bloodstream, lipid distribution occurs over a longer time frame compared to water-soluble nutrients. This extended delivery contributes to the regulation of energy availability and supports sustained metabolic activity. When formation is consistent, lipid supply is steady. When it is disrupted, delivery becomes irregular. Chylomicrons therefore serve as the link between intestinal absorption and systemic lipid distribution. They convert absorbed lipid components into a transportable form that can move through the body without disrupting fluid balance. Their formation and function depend on the alignment of upstream digestive processes, and their effectiveness reflects how well those processes have prepared and delivered lipid substrates to the point of absorption. Nutrient allocation versus simple absorption. Absorption describes the movement of nutrients across the intestinal membrane, but allocation determines where those nutrients go, how quickly they are used, and whether they are stored or oxidized. These are not the same process. The intestine can transfer substrates into the body, while allocation pathways decide their fate within circulation and tissues. A system can absorb nutrients effectively and still distribute them in ways that do not match current demands. The outcome depends on signaling, transport structure, and the condition of the receiving tissues. Water-soluble nutrients enter the bloodstream and are distributed according to concentration gradients and tissue uptake signals. Glucose and amino acids move rapidly through circulation, and their allocation depends on hormonal signals that control uptake and storage. When tissues are primed to receive these substrates, transport into cells proceeds efficiently. When signaling is altered, these nutrients remain in circulation longer or are redirected towards storage pathways. The presence of nutrients in the blood does not guarantee their use. Allocation determines whether they are oxidized for energy, incorporated into structural components, or stored. Lipid-derived nutrients follow a different route that reflects their physical properties. Chylomicrons carry these lipids through the lymphatic system and into circulation, where they are gradually delivered to tissues. This stage release creates a delayed and sustained allocation pattern compared to water-soluble nutrients. The distribution of lipids depends on tissue demand and enzymatic activity that governs the release of fatty acids from transport particles. When this system is aligned, lipids are delivered in a controlled manner that supports membrane structure, energy production, and signaling functions. Allocation is influenced by the condition of the digestive sequence that precedes it. When digestion and absorption produce consistent substrate forms, transport and distribution follow predictable patterns. When upstream processes are irregular, the timing and composition of absorbed nutrients vary. This variability carries forward into allocation, where tissues receive substrates in inconsistent amounts or at inappropriate times. The system adapts to these fluctuations, but the stability of distribution is reduced. The distinction between absorption and allocation becomes clear when considering capacity. Absorption capacity is defined by transport systems at the intestinal level, while allocation capacity is defined by how tissues receive and process incoming substrates. If tissues are not prepared to use a given nutrient, it may be stored or remain in circulation. This can occur even when absorption is functioning normally. The system must align both processes to maintain efficient use of nutrients. Hormonal and neural signals coordinate allocation by linking nutrient availability to tissue demand. These signals adjust uptake rates, storage pathways, and energy utilization. When they are aligned with the actual condition of the body, allocation supports stable metabolic function. When they are not, nutrients are distributed in ways that do not match current needs, leading to inefficiencies in energy use and structural maintenance. Nutrient allocation, therefore, extends beyond the act of absorption. It determines how the products of digestion are integrated into the body's systems. Absorption moves substrates into circulation, but allocation governs their distribution, timing, and use. The effectiveness of this process reflects both the quality of upstream digestion and the state of the tissues that receive these nutrients.